Amazing. I'm really happy to be here and to present the challenges and solutions to unravel molecular mechanisms of biomolecules. And so what I'm going to do is I'm going to start with introducing our team, the structural and molecular biophysics team. Then I'm going to present to you an overview of the biological systems that we're interested in, and what are the timescales and resolutions that we're interested in resolving, and also what are the observables, the key observables that we want to measure. Then I'm going to present the challenges that we face in studying these biological systems, and I'm going to present a method that we've been developing to overcome one of these challenges using cryo-electron microscopy. And I'll finalize with the conclusions of our work. So the structural and molecular biophysics team is a new collaboration just started last year between CCM and CCB. Sonia Hansen is on the CCB side, and I am on the CCM side, and this is an amazing group of scientists that have started this collaboration and looking to solve problems in structural and molecular biophysics using both experimental and computational techniques. So the biological systems. We're very interested in proteins because they are the nanomachines of life. These are the working force. And so a protein is a chain of amino acids. And so these amino acids, there's 20 natural amino acids. And these building blocks have been around for more than 3 billion years ago. So for a very, 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 very long time, we have always used the same building blocks to construct and basically build the working force of nature, which are proteins. And so what we're interested in is trying to understand how these biomolecules work, both using experimental techniques and simulations. So a little overview about the scales in biology. So this is a light microscopy image of a cell, and this is around 2 micrometers long. So if we zoom in, we need a different microscope. This is a TEM, transmission electron microscope image, and this is around 1 micrometer long. So here you see different structures within the cell. Now, to study proteins, we need to zoom in even further, of the order of 10 nanometers to 100 nanometers, say, in size. But we're interested in visualizing these proteins at atomic resolution. So one of the things that we're very interested in is in resolving the structure of proteins and how the atoms rearrange in 3D space. So even if we have systems that are of the order of 10 to 100 nanometers, we really want to see how the atoms arrange in 3D space. However, something important is that we're not only interested in static structures, but we're also interested in the evolution of these atoms, how the atoms evolve in time. And so here I wanted to show you a little bit about the biological timescales that we're interested in. So up here we have the fastest timescale. This is a dihedral angle rotation, and it's of the order of 1 nanosecond. Then here we have phenomena that are, for example, the diffusion of ions through channels of the order of hundreds of nanoseconds to milliseconds. Then over here we have the unbinding of a drug to a protein active site. And this unbinding takes of the order of microseconds to seconds. And for example, here on the right, we have, this is the SARS-CoV-2 spike protein, and here we have the RBD domain. And I'm going to talk to you a little bit more about it in the next slide. But it opens up, and this opening is of the order of 1 second. So basically, we're interested in phenomena that range in a really, really long scale from nanoseconds to milliseconds, microseconds, and to things that take days. So it's a very wide range and challenging range.
What is RBD?
So the RBD is a domain Here we have it. So it's this domain here, and it's the name of this domain of the protein. So basically a specific part of a protein, of the spike protein.
Just the receptor binding domain?
Receptor binding domain. Okay, so how are we going to study these biomolecules? What are we going to extract? What are the observables that we're interested in? So this is the spike protein, and what we're interested in this biomolecule, its time evolution, but also in a native-like environment. So what we're interested is, this protein is normally surrounded by water. It's also in contact with the membrane. So how does all of the environmental conditions also change its biological function? And so here we have systems that are of the order of, for example, 750,000 atoms, if we include the surrounding water molecule. So it's really, really large. And because we do not have an initial condition measurement, it's all basically distributed by Boltzmann factor. What we're interested is in looking at two main observables. So the first observable are the probability distributions of states of the configurations, and I'm going to talk a lot about this. But basically, for example, in this case, this domain, when it's closed, when it's in this configuration, the spike protein and the virus protein is shielded from the human immune system. So basically, it's undetectable. And then, but it's the first contact. When it opens, it makes the first contact with the human receptor. And so this is the first point of entry of the virus into the cell. And so you have this spike protein in two states. It's either a closed state or an open state. And so we're interested in extracting these observables from this very complex system. And what we want to know is, what is the probability of this domain to be in the closed state or the domain to be in the open state, and what is the dynamics between the two states. So what is, for example, the average transition time to go from one state to another state? And these are basically let's say the ensemble properties that we can measure with these systems. So a little bit more about these observables. So the free energy is related to the probability distribution of configurations by a Boltzmann factor. And so here is an example of a free energy surface. And so this is the global free energy minima, so it's the most probable state. So this could be thought as the folded state of the protein. And then we have other metastable states of the system. And so we're interested in comparing these different metastable states of the system. What is the difference in their free energy? And we also want to learn what are the barrier heights. How much energy do I have to put into the system for it to transition from one state into another? So this would be like, say, a static picture of the probability distribution of the configurations of a biomolecule, but we're also interested in the dynamics. In the average time, for example, to go from this state to jump to this state. And this has to do with, for example, the diffusion coefficient and the dynamics, the underlying dynamics of the system. And so we want to, overall, once we extract these observables, we will understand a little bit more about how really the biomolecule performs its biological function. So why is this important? So as I mentioned, it's important to understand the biological mechanism, but also, for example, once we have these observables, we can understand what happens with mutations, what happens with variants, how do mutations alter these biophysical properties. Or, for example, something that Sonja Hansen is very interested in is, how does temperature change these landscapes and changes the dynamics? Or, for example, we're interested in trying to understand how small molecules, for example, drugs, alter these conformational landscapes, these conformational states. And so this is relevant for applications, say drug design, synthetic biology, but it's also relevant for basic science. So really understanding, for example, how biological temperature sensing works. And so here, this is basically, I showed you the overview of the systems that we're interested in and the observables that we want to extract. Now I'm going to present to you what are the challenges in studying these systems. So on the experimental side, we have structural biology experiments, and here we have X-ray crystallography, nuclear magnetic resonance, and small angle X-ray scattering. And so all of these measurements are ensemble averages. What does this mean? You have multiple copies of your protein, either in a crystal or in solution, and then you run through these systems some type of measurement, and you have this measurement is averaged over the ensemble of proteins that you have either in solution or in your crystal. So basically, the problem and one of the issues is that really these ensemble averages limit the extraction of single molecule information. So it's very difficult or almost impossible to extract a free energy landscape or to extract dynamics from these structural biology experiments. Okay, so now on the other hand, we have another type of experiments, biophysical experiments, and these are called single molecule experiments. And they're very interesting because you can measure things over individual molecules. So for example, in force spectroscopy experiments, we have these optical tweezers, these beads, and these beads are attached to a single molecule. And so what you measure in these experiments is the extension as a function of time. And so you would see a trace that is similar to this one here. You would see that the extension goes down, for example, the green one, then it increases, then it goes down again, and it fluctuates stochastically as a function of time. And so something similar here is, for example, with these FRET, single molecule experiments, you have two molecules that are attached to your protein, and when they are in close contact, they emit a different type of light. And so you can also basically probe distances between some extension of your molecule. You can probe these distances with a single molecule FRET. So
what does FRET stand for?
So it stands for Foster Resonance Electron Transfer, I think.
Okay, right.
And I think actually there's another name for it. And so these observables really prone of showing how the biomolecule changes, how the biomolecule fluctuates in time. But one of the issues is that these observables are very low resolution. So it's basically only a distance, whereas we would, over something that has, 10,000 atoms, you can only measure one single distance, or maybe like nowadays you can have 3 distances in FRET. But really having like seeing the evolution of the atoms is not possible because you're projecting down over a variable that isn't great. So the take-home message on the experimental part is that there's no single experiment that can give us all the information that we would really like. There is a trade-off between the spatial and the time resolutions. So either you have structural biology experiments that are really good at resolving atoms, but static and ensemble averages, or you go to the single molecule ones that are good at the dynamics part, but you lose the information of the atoms. Okay, so this was on the experimental side. Now on the simulation side, as I mentioned before, we want to study the biomolecules in native-like environments. And basically, there are 2 things that are important. And what we would like is an accurate description of the interactions and we would also like to see the system's evolution for biologically relevant timescales. And so, this is basically going to be another trade-off. And one of the main questions that we asked ourselves, like the community asked themselves, was, okay, to what level of description, what is the level of description that we want to describe the main interactions in biology? So if you have a system with 1 million atoms, you can think, okay, well, I'm just going to make big blobs, concentrate a lot of atoms, and group these atoms in a coarse grain-like fashion, and try to basically solve some equations on top of these coarse grain-like blobs. But one of the issues is, and basically the problem is, I would blame the hydrogen bond. So a hydrogen bond is a no-covalent bond formed by a dipole-dipole attraction between a hydrogen and an electronegative atom. And hydrogen bonds are essential in biology. These are the reasons for why water has all of its amazing properties. And also, these basically are the backbone formation of the secondary structure elements in proteins, alpha helices and beta sheets. So all of the many key interactions in biology are dominated by hydrogen bonds. So basically, we want to take this into account, but this means that we require explicit hydrogens in our simulations, and having hydrogens is going to limit our simulation time step. And so basically, because these are small and they vibrate very quickly, this is going to be our maximum time step that we can have in our simulations. And this is going to be like one of the trade-offs that we're going to have. So there are many systems and like in biology that require higher level of accuracy. For example, enzymes where you have breaking of covalent bonds, so you have transfer of electrons, and you need quantum mechanical calculations for these. But there are another very, very large set of biological systems, like the ones that I'm showing now, that do not require explicit treatment of the electrons. So basically, what we're going to do is we're going to concede on this level of accuracy where we're going to have explicit all the atoms of our system, but we're going to treat them classically with molecular dynamic simulations. So we are going to have basically a description is a classical potential, and this classical potential is going to be divided in two main terms. So one is the bonded interactions, and so in these bonded interactions, our atoms are going to be represented as spheres, and bonds and angles are going to be represented as springs. So in this force field that we have these bonded interactions. Basically what they do is they make the molecule have its chemical shape and preserve its chemical geometry. But this is basically invented, but it's been validated with QM, quantum mechanical calculations, and also experimental techniques. And we use these classical potentials basically because having a wave function is too expensive with the number of atoms that we have in our system. And so the second part in the force field are the non-bonded interactions, and there are basically two, a Coulomb interaction and a van der Waals interaction, that represent the electric statics. And so these force fields have been developed for the last 40 or 50 years. There's many types of different force fields, but they've been particularly optimized for water and proteins, which are amino acids. And so then what we do is we have this potential, and then we numerically integrate Newton's equations with the time step, the longest time step that we're allowed, which is of the order of 1 femtosecond. And so we have to integrate Newton's equations numerically many, many times to see the evolution of the atoms of our system. And so with classical molecular dynamics, what we have is we have an initial configuration, and then we start calculating the forces over our system, integrating them Newton's equations numerically, and seeing how our system evolves in time. But there's a problem, and the main problem is that we want to reach these time scales that are really, really large. We want to see phenomena that are of the order of milliseconds, of the order of seconds, having millions of atoms in your system. So it's really, really challenging to reach biological relevant time scales with classical molecular dynamics. And in many cases, with the computational resources that we have now, it's impossible. The vibrations of the hydrogen atom.
Could you use your seat mic, please?
So you really have to resolve the vibrations. I mean, that's what's going to make it, change its shape. You can't. There's no other.
So there are some algorithms that try to trick this a little bit, but in the end, you might get 2 femtoseconds, 5 femtoseconds maximum. But still, if you have an all-atom representation of your system and these small bonds, that it'll basically be around that order of magnitude, that the femtosecond time step that you can get. Yeah.
Can I follow up as well? I mean, let's say we got rid of the hydrogen and went to the next, like the carbon backbone. So those ones are a bit slower, but it's still a few femtoseconds. Is that what you're saying?
Yes. So I actually don't remember correctly, but I think it would be. So maybe if we took away the hydrogens completely and we didn't have completely stiff bonds, I think you could go to 10 femtoseconds. That would be my guess.
Yeah.
But this was thought about a long time ago when the force fields were starting, and then things stayed like that.
Yeah. It is kind of, I mean, we study ODEs and ordinary differential equations, and when they're stiff like this, sometimes you can kind of forget the little oscillations and go over much longer times. But here, the thermal oscillations are really actually important. Is that it? You can't get around that.
I think so. This would be my guess.
Yeah.
Yes.
It's interesting. Okay.
Okay. So with biomolecular simulations, we have the same problem. There's a trade-off between the accuracy that we want, which would be ideal. We would like to have everything QM, right? But we can't. And the simulation time, how long we can simulate for with the computing resources that we have.
A question on Zoom, actually, from Jinpeng Wu, who says, he says the timescale is large. What kind of problem will it make in simulate? Okay, I guess how long can you actually simulate for? I think he's asking. And then he's saying, is the computation, is it CPU, is it flops bound or memory bound? Or what's the limit on this?
So this depends a lot on the number of atoms in the system that you're interested in. So if you have a small globular protein, say, surrounded by water, this would be around 100,000 atoms. And so for something like that, you could go up to seconds, probably but really, really investing a lot of computational resources. That would be the maximum. I think I would say nowadays the largest simulation that I recall is one of the spike protein, the large protein that I showed you, around 700,000 atoms, and this reached 1 second in folding at home, which is this grid-based computational resources, which is that's basically yeah, more or less what we can do, but that sometimes is not enough. The problem is that these are all stochastic events. So when you see you start from an initial condition and you let it run for 1 second, it doesn't mean that it will always do that, right? So you have thermal fluctuations that depending on are stochastic. And so if you run the same thing with different initial velocities, you will get a different trajectory, so you need to do this many, many, many times to really have some ensemble or statistical property.
About this, so how do you choose these initial conditions? How do you choose? I mean, for the position,
Yes,
I guess you know them. But for the velocity distribution?
So normally what we do is we rely a lot on the structural biology community to give us good initial structures, and we start with good initial structures. If there's not a resolved structure, then probably we would go to something like AlphaFold or some homology modeling of the protein. And then we would typically run many simulations, replicas of your system, starting from the same structure with different initial velocities. And then look at the properties of these different replicas and calculate averages of the observables.
Is there a specific distribution you choose for the velocities? Or
so it depends on the thermostat that you have. Okay. But it's some random of the thermostat that you follow.
Okay.
You mentioned that if you don't have a structure ahead of time, then you use AlphaFold. Is that the state of the art?
I think so. I would say yes. But I don't know. Who knows? Like, I don't want to go into this discussion.
Okay. Sorry.
Or Rosetta, right? So.
A question from Andreas Buja as well, but you can say that, yeah, just briefly saying how do you handle rare events when there's crucial but rare events? Yes. Is there a way to guess at those?
No.
So you don't have to simulate the thing for a long time.
Andreas, that's a great question. So basically, all of biology is dominated by rare events. We have gigantic, so all of these like timescales and the phenomena. It's basically you're trapped in a local minima for a very, very long time. Nothing happens, and then suddenly everything happens, for a very short time. So we can't. So it depends on what you're interested in. So for example, I'm very interested in like probing these transitions, and so what I do is I normally look at possible degrees of like motion where they should occur. I call these collective variables, and then I bias the system towards this direction. But this is basically another complete topic that it's for another presentation. But you can push your system over the interesting degrees of freedom. Okay, good. So now what I'm going to do is I'm going to talk to you about one method that we've been developing in the group. And it's related to cryo-electron microscopy and basically the first set of challenges related to experimental techniques. So as I mentioned before, we have X-ray crystallography, NMR, SACs that are basically ensemble averages, ensemble average experiments. But cryo-EM is really, really interesting and very particular, and now this is what I'm going to talk to you about. So in cryo-EM, we have a transmission electron microscope, and what we have is our sample, which is copies of the same protein, the protein that we're interested in, many copies, that are in different orientations in the sample and unknown conformations. And so what you do is you basically put your beam of electrons through the sample, and then you have these images. And so what cryo-EM has been focusing on is using these 2D projection images to reconstruct a 3D density map of the molecule. And so basically, cryo-EM has gone through the resolution revolution, where in the last 10 decades, there has been great advances in the electron detectors, so in the cameras that you detect the electrons, and also in the processing algorithms. And nowadays, it's possible to have really, really high-resolution structures, high-resolution maps, using cryo-EM. So here, what I'm graphing is the yearly average resolution in cryo-EM over the last 20 years, more or less. And so you see that, like, in the last 10 years, there's been a massive drop or increase, increase in the resolution. And here, what I'm graphing is the number of PDB structures deposited in the PDB. And so you can also see that many more people have been using cryo-EM to resolve structures. And this is basically because many biological systems, for example, membrane proteins that were very difficult to crystallize, or large protein complexes that it's not possible to resolve with NMR, have been resolved with cryo-EM. And so because of this, in 2017, the Nobel Prize in Chemistry was given to Dubochet, Frank, and Henderson, both on the experimental side and on the computational side. And even recently, like, cryo-EM still surprises us, and it was possible to get in 2020 a structure of apoferritin of 1.2 Armstrong resolution. So this is like pinpointing the atoms, which is really cool. Now, why are we interested in cryo-EM? We're interested actually for another reason. And the reason is because cryo-EM is a single molecule technique. So in cryo-EM, what we have is we have our protein that can adopt different conformations. And the freezing is done very rapidly. It's done of the order of microseconds. So any conformational transition that is slower than that will be trapped in the different conformations. And because we're taking images of individual molecules, it's possible to trap them in different conformations, and these in principle should be distributed to Boltzmann at the temperature prior to freezing. So there's much more information than just an average. And so basically, this might be a better impression of how we see really biology works. So we're taking pictures of a horse running. And then you take your pictures, then you basically throw them randomly, and then these are in random orientations, right? So that you have your random orientations and your unknown also conformations. Now, how does cryo-EM deal with conformational variability? Well, what is this? This is a typical example. And so what is done is that you start with your sample and you pick your particles, and you have around 1.2 million particles. And then you do 3D classification. And so basically, so very, very nice techniques have been developed to classify out your particles into different classes, different 3D classes. And so you start generating maps from these different classes, and then you start filtering out particles, and then you arrive with a set of particles that gives you a very high resolution density map. This is more, like in this case, it's less than 15%, but really, like, you're throwing away more than half of your data. And one of the reasons, like from the experimental perspective, is that so when you bombard your sample with electrons, the sample suffers radiation damage. So many of these particles can be basically destroyed or broken proteins, and we're not interested in broken proteins. And so what we want is things that are intact, that are solid, and that give us a high resolution structure. From my perspective, what I think is that there's definitely conformations, different conformations that are being thrown out. So probably not all of them, but probably a good chunk. And so basically what the cryo-EM field, what it does is it says, okay, I'm going to focus on this conformation, and then I'm going to classify them out and just stick with the images that give me the high resolution structure of this conformation. Okay, so one of the questions was why discard so much data? And like a more like fundamental question is, can we extract free energies? Can we extract probability distributions from the cryo-EM images? And so, so what is a free energy? Well, say we have this a configuration that lives in this very, very large space. N is the number of atoms. And so the Boltzmann factor gives us the probability, so this would be the probability of this configuration, and U is the potential energy. So this lives in this very high dimensional space of 3 to the number of atoms. And so what we typically do is we project down along some degrees of freedom or some collective variables. And so basically, the free energy here is related to the probability distribution along these collective variables. And so now, basically, by projecting it down, we can have a picture that we can try to understand, and then we have information of the global free energy minima, the metastable states, and the activation barriers of the system. Okay. So, given the large number of work like being done in cryo-EM, it's a little bit surprising that not so many people have worked on extracting free energies from the cryo-EM data, but there are some groups. And one of the main groups, and like this is basically the pioneering work, is by Joachim Frank, which is one of the Nobel Prize laureates. And what they do is they use machine learning and diffusion maps to basically map the particles on a low dimensional space and calculate the free energy surface of, the first work was on the ribosome. Other people from Holger Stark's groups mainly have done principal component analysis to basically define the space and then try to map where the particles are. So there are two questions that we asked ourselves here, because so basically what these groups did was they just analyzed the data and they showed free energies, but like, is it really possible to extract free energies from the cryo-EM data? Nobody had shown it. And the other question was like, how are we going to filter these particles? Because before, with the like standard techniques, you would just throw them out. But now if we're looking at probability distributions, we have to be careful. We have to count, and all the particles count, and will give us basically the normalization in the probability. So how can we weigh or filter out the particles?
Can I quickly ask a question? You said that the electrons can actually sort of break the proteins. So, like, are we sure that the electrons aren't going to affect the conformations of what we're seeing?
No. So that's one of the reasons why we freeze the sample, to make it like more solid and less affected by the beam. But they do. There's, for example, so the main advance in the resolution revolution was that, so they changed the detectors, and they could basically now measure directly where the electrons reached, and they could also measure it in different time slots. So they could do something that was like they called take movies. And so what you would see was instead of one single image, you would see one image, then another frame, then another frame, then another frame. And when they looked at these frames separately, they actually realized that the electrons would move in the first frames. They would really displace the particles. And so what the direct electron detectors gave us was really a movie of what happened in this heating process, and you could correct for that. So they corrected for the beam-induced motion, and that was one of the main keys of going toward the resolution, the revolution, because they could basically increase the signal because you're not averaging over all of this thing that was induced by the electrons, but you could basically classify the movement induced by the beam.
So there's a question from Yasmin on Zoom, and she asks a tough one, which you're going to enjoy. How are the resolved structures / maps from cryo-EM verified?
So this has been a topic of discussion. So what is typically done is something that uses the Fourier shell correlation, but basically, so you take your particles that you think are good, and then you divide this into two random sets, so two separate sets of particles, and you generate reconstructions from the two sets, and then you compare these reconstructions, and you basically analyze the correlation in Fourier space. And basically up to where the correlation is higher than noise, then you say, okay, that's my resolution. The problem is that, so here, so these images are really, really noisy. So you have of the order of 100 times or 10 times more noise than signal. So you can align noise very easily. And so when you do this reconstruction process, you are basically biasing your system toward some that depends on initial conditions and stuff. And so if, in many cases, even if you do this process, you can still make Einstein from noise. Basically, this is known in the field that you can basically generate things that are not there just because of alignment processes. So it's still tricky and not completely resolved an issue. Okay, so statistical mechanics and free energies is basically the art of counting. And so what we want to do to extract the free energy is say that you have a low dimensional space that maps to your configurations. And what you want to see is, I have my image, the cryo-EM image, where does it go? To which configuration, where does it go? And then my free energy is basically an histogram, or the negative log of the histogram of how many particles are where. And so it's basically an art of counting. And so here I just wanted to present and outline this cryo-EM Bayesian inference of free energy profiles that we've been developing here at the Flatiron. And so this has been a very nice collaboration with Alex, Bob, and Eric. And so this is inspired by methods from the MD community. And so what you have is, say that the surface is your high-dimensional space where your protein lives. And so what we're going to do is we're going to build a path that takes me from one relevant state of the system to another relevant state of the system. And so here, as an illustration, for example, I can build a conformational path that goes from a closed state of an ion channel and takes me to an open state. So what you can imagine is, you can imagine basically this trajectory is a movie that goes from the system like this to the system like this. And so then here we have our cryo-EM particles. And basically the main question is, okay, where do these cryo-EM particles map to? Like, are they coming all from this state? Are they coming from this state? And basically, can we extract a free energy profile? So some free energy profile along this path that will tell me that will relate to me the probability distribution of these configurations along the path given the cryo-EM images. And so what we did was we proposed this Bayesian formalism to calculate a posterior probability of a free energy profile given a set of particle images, cryo-EM particle images. And so just in essence, what we do is say we're only looking at a single cryo-EM particle. And so what we have is we have our 3D configurations that go from one state to another, but this is, you can imagine it's discrete. And so what we do is we calculate a likelihood of that image being generated by one particular model in the path. So what is the likelihood of this image being generated by this one, being generated by this one? And so how we do this in practice is we take the three-dimensional structure, we generate synthetic images from this 3D structure, and then basically we compare the experimental image to all those synthetic images generated by this 3D structure. And so we can calculate this probability of that image given a free energy profile by this expression here, which is basically this likelihood for each model along the path and the Boltzmann factor of the free energy along that particular point in the path. So for multiple images, we take them as independent samples, so we just take the product of the single images, and we also have a prior along the smoothness of this free energy along the path. So what we do in practice is we're going to sample many G's, many profiles, given this posterior probability. So if we have this, say, underlying free energy, what our method would do, we would generate many MCMC samples, for example, using the posterior probability, and then we can calculate the mean and the uncertainty of this free energy profile. So here are some results. So we first worked with a system that was completely under control with synthetic data. And so this is the HSP90, and it has one movement, 1 degree of freedom, this basically arm that opens and closes. And what we do is we generated synthetic images from that system with a wide range of cryo-EM conditions. So different signal-to-noise ratios, different number of particles, different orientation directions, and with a known free energy profile, known distribution along the models along this path. So we basically have everything under control. And our question is, under which conditions, under which cryo-EM conditions can we really recover the underlying free energy? And what are basically the challenges when you're trying to map 2D images along this path? And so the first thing that we found is that the projection direction, estimating really what is the direction where the image was projected is crucial, and you need it to be, you need a refined grid in the orientation search to be able to have a good free energy. So here the black line is say the ground truth, that what we generated the particle distribution from, and the orange curve is the estimate, the extracted free energy using our method, but having a very coarse grid on the orientation search. So basically our orientation accuracy is not great. And so you can see that we're relatively okay with the difference in the minima, but the free energy barrier is underestimated dramatically. Whereas if we have a better orientation accuracy, which is the blue curve, we can recover almost even the barrier and the difference in the minimum. So basically the take-home message is that the orientation direction and the accuracy is important. And the second factor that we found that is very important is the signal-to-noise ratio of the particles. This is more an experimental condition. And so here we have the same underlying free energy. And the green curve is going to be particles that have very low signal-to-noise ratio. So signal-to-noise ratio that is about, 1,000 times, noise that is 1,000 times larger than the signal. And so basically for this set, we cannot recover the underlying landscape. But if we have images that have, say, 100 to 10 times more noise than signal, which we consider high signal-to-noise ratio, then we can recover, at least for this, say, relatively simple case, the underlying free energy landscape. And we can estimate very well the barrier. So something interesting is that if we group all the particles, so those with low signal-to-noise ratio and high signal-to-noise ratio, which is the blue curve, we actually obtain a relatively good extraction of the barrier and the minima. And the reason is that basically in this Bayesian analysis procedure that we're doing, it naturally reweights the contribution of each particle. So particles with low signal receive a lower weight. Okay, so this is the final system that I wanted to show you. This is on real data. And so this is an ion channel that is activated by calcium. So you can think about these yellow spheres as calcium bound. And it actually was resolved in two states, the calcium bound state and the calcium unbound state, which are different. And so this was resolved in two different samples. So there was one sample that had a lot of calcium inside, and there was another sample that didn't have any calcium. And so what we did was we took the sample, all the particles from the calcium bound sample, or the sample that had calcium, and we asked ourselves, is it possible that there is, because we're in a thermodynamic equilibrium, is it possible that there is some of these particles in this state without calcium? Can we recover? Can we look for this in the data? So this was the first question, and the second question was, can we extract a free energy profile? And so basically what we did was we generated our path of transitions. So each time it goes down, you can see it just so 0 would be when it's down, and 1 would be when it's up. It's repeating the cycle. But so basically we're going from 0, we're going from the calcium bound state, and 1, we're going to the calcium unbound state. And so we ran this with our method and our real cryo-EM particles, and we found that the calcium bound state is the global free energy minimum. This makes sense because this was the cryo-EM particles that were used to generate the calcium bound state. It's the data set with calcium. But interestingly, we found a metastable state that was the calcium unbound conformation, and all of these particles were thrown away. We were very grateful with the experimentalists that they actually put in all the data, and they discriminated between the ones that were used to generate like the high resolution structure. But we could find in the data things that basically were discarded. So we're very excited about this method, and now we're thinking about many things. We're thinking about can we optimize the path, because the path that we're generating, these conformational transitions, are basically invented. So can we find the minimal free energy path given the data, optimize the configurations to basically have a biological relevant path transition? And also we're thinking about, what about cooling? Like we flash freeze, but what does this mean in terms of how the free energy landscape changes and how can we correct for this? We're thinking about validation methods, so we're really excited about this, basically this method. And so finally, I would just, the take-home message is biophysical experiments and simulations are governed by a trade-off between spatial and temporal resolutions. And in our team, what we're trying to do is we're trying to basically bridge bridge this gap to understand how biomolecules work using both theoretical and computational algorithms. So I would like to acknowledge the structural and molecular biophysics team. It's been great to work with Sonia and with everybody. Also, all of our collaborators in CCM and CCB, Leslie and Mike, the admin and the SEC for all their help, and thank you so much for your attention.